Hello and welcome everyone. I'm Katy Kettleborough. I'm a member of the CF AMR Syndicate management team, and I'm also the leader of the LifeArc Chronic Respiratory Infection Translational Challenge. So we're running this webinar today to coincide with the launch of the Syndicate Collaborative Discovery Programme. And this is our first funding call for novel antimicrobials to treat lung infections in CF. So just to give you some context of what we're going to try and cover today. We're going to have a background from Paula Sommer in a moment. We will then have a short film from Angelo Micciche, who is someone who lives with cystic fibrosis. Professor Jane Davies will then set the scene for us. And Jane is Professor of Pediatric Respirology and Experimental Medicine at the National Heart and Lung Institute. She's also an honorary consultant in Pediatric Respiratory Medicine at the Royal Brompton and Harefield. And as I say, Jane is going to set the scene for us. And then we'll move on to Ed and Neill, providing an overview of the funding call, and that will include scope, eligibility, the sorts of things that we want to be able to offer as part of this collaborative program. So without further ado, I'm now going to hand over to an introduction to the syndicate.
Hello, everybody. I'm Paula Sommer. I'm from the Cystic Fibrosis Trust. The Trust is a UK-wide medical research charity that funds CF research and advocates for people living with cystic fibrosis. So cystic fibrosis is a recessive, genetic, life-limiting condition that affects over 160,000 people worldwide. Although cystic fibrosis affects the whole body, people with cystic fibrosis are susceptible to lung infections. And once these adapt to the unique environment that is the CF lung, they can be difficult to treat, and repeated infections can cause permanent lung damage. People with CF necessarily are reliant on antibiotics, and so antimicrobial resistance is frequently seen and is a cause of mortality. So antimicrobials and related diagnostic tools remain a key and very necessary therapeutic in cystic fibrosis. And key CF pathogens are included on the World Health Organization priority list. So although we're very fortunate to be in the era of highly sophisticated CFTR modulator therapies for many, but not all people with cystic fibrosis, infection still remains really important to the CF community. The Trust recently, in partnership with other organizations, supported a refresh of the James Lind Alliance patient priority settings for research in cystic fibrosis, and infection and antibiotic resistance featured in the top 10. So because this is of such concern and importance to the CF community, the Trust seized at the opportunity to partner with Medicines Discovery Catapult to form the Cystic Fibrosis Antimicrobial Resistance Syndicates. And we were both really delighted to welcome LifeArc as a third managing partner last year. The mission of the syndicate is to accelerate the translation of new antimicrobials and diagnostics for CF lung infections through collaboration, bringing these to the clinic faster for the benefit of people with cystic fibrosis. So our aims are to provide guidance on the medicines discovery roadmap for lung infections and make sure that these align to the needs and priorities of people with cystic fibrosis. To catalyze the development of and access to specialist enabling research resources that will help innovators, to facilitate the formation of virtual medicines discovery teams and R&D collaborations across the network, and lastly, for the purposes particularly of today, to enable innovators across the globe access to specialist support and funding to support diagnostic and antimicrobial projects for lung infections and cystic fibrosis. The unique value of the syndicate lies in its very diverse stakeholders, managed by Medicines Discovery Catapult, Cystic Fibrosis Trust, and LifeArc. and anchored by people with CF and their loved ones, whose involvement activities filter across all of the syndicate activities. The syndicate brings together leading academic and industry experts, clinical teams, funders, investors, translation and commercialization experts, all working together to accelerate the translation of new CF antimicrobials and diagnostics, bringing them to the clinic as quickly as possible. So when the syndicate first started, we reached out to innovators in the space to try and identify the biggest challenges. And two key themes emerged. Firstly, the access, the lack of access to clinically relevant strain samples, isolates, and data. And secondly, the lack of an agreed preclinical testing pathway. And so the syndicate decided to develop a suite of resources to try and address these. So the first of these was the UK CF Infection Biorepository. So the biorepository is a virtual network that joins together centres across the UK that have unique collections of strains, clinically relevant strain samples, isolates, and associated data. The biorepository is accessed by a single access point, which will hopefully make it easier for innovators to access. To address the lack of a preclinical pathway, the Trust and Cystic Fibrosis Foundation came together to fund a project at the University of Liverpool, led by Professor Joe Fothergill, that is around developing an evidence-based preclinical framework for the development of CF antimicrobial therapeutics. And then the syndicate joined together to develop a set of very patient-centered, patient-focused target product profiles for the development of antimicrobials against Pseudomonas and Mycobacterium abscessus infections, which we'll hear about a little bit later, and the syndicate's pursuing a similar route with diagnostics. The syndicate's also developed a CF AMR network, and the network really aims to bring together stakeholders across the board to come together to disseminate information, to facilitate knowledge exchange, and to catalyze collaborations. And if you're not a member of the network, please do think about signing up. Syndicate continues to grow, and we plan on continuing adding to our toolkit of supporting resources for CF antimicrobial and diagnostic development. We also plan to grow and nurture the cross-sector CF AMR network. And very importantly, for the purposes of today, we also plan to identify, enable, and support CF antimicrobial and diagnostic programs. And today you'll hear about one of our antimicrobial programs, the Collaborative Discovery Program. But first, before we do all of this, let's just remember why we're here today and listen to a personal perspective on living with cystic fibrosis.
I'm Angelo, I'm 48, about to turn 49 next week, and I have cystic fibrosis, dealing with the tiredness and the night sweats and, you know, all of that constant coughing and being short of breath and just not being able to do all the normal things that everybody else would do and take all that medication, which obviously has been huge really as time has gone on. It's just increased and increased, so to the point that I'm, you know, 60-70 tablets a day now, and my choice of IV antibiotics that's left is really limited. Probably got about 9 or 10 that I cannot take anymore. I'm 2 years into using modulators, and you know, I still have IVs treatments, you know, probably 2-3 times a year at the moment. So it still has that, you know, CF has not gone away for me. The prospect of, you know, new antibiotics or new tests, or, you know, to detect, you know, infection at an earlier stage, for example, would be amazing. Or, you know, anything that improves quality of life, I think, would be absolutely, you know, transformative, I think, for people with CF.
Thank you for that. I'm now going to hand over to Jane Davies, and as well as the roles that I'd already highlighted that Jane has, she acts as the Deputy Chair of the CF AMR Syndicate Steering Committee, and she's going to talk us through her clinical perspective on the need for new antimicrobials in CF and what good looks like in terms of the target product profiles. So over to you, Jane.
Thank you very much, Katy, and thank you, Angelo, for really sort of, I think, bringing this issue to life for people. Angelo mentioned CFTR modulators, which are drugs which have really achieved fantastic progress over the last decade or so. But I want to really use this to illustrate the continuing problem of lung infections despite these being available. I won't go through the science behind cystic fibrosis in great detail, but you can see here within the cell, there are a number of ways in which the CFTR protein can fail to be produced. And the understanding of the biology to that has led to these small molecule oral drugs, which can either improve the function of the protein once it's at the cell surface, or more commonly, they can actually help the protein get to the cell surface, and then with a dual action also improve the function. The first one of these drugs that we had available targeted this group here of patients who have a sort of mutation in their gene which leads to protein being in the right part of the cell but doesn't work properly. And that was the low-hanging fruit really in terms of the cystic fibrosis population. But it was only about 5% of the population who were suitable for this first drug to come along. But the fact it's been with us for about 10 or 12 years now means that we can start to look at long-term outcomes. And what I've shown here is just some 5-year data from 2 registry data sets, so large groups of patients from US and UK. And you can see the widening gap in those bars between the grey and the black as people experience life on this particular modulator, and the frequency of chronic Pseudomonas infection does appear to be impacted. But I think you could probably agree that that impact is relatively modest. Now, as I said, that's only 5% of the population. What we really need to do is to target the commonest mutations in CF, and that requires a more combined molecule approach. And this is a very, very exciting triple molecule that we now have available to us in the clinic for people aged 6 years and older, of course, elexacaftor, tezacaftor, ivacaftor, we'll call it ETI, which gives really great improvements in lung function. And you can see here down a decrease in the frequency of exacerbations leading to hospitalization or needing intravenous antibiotics. But what we don't know is the impact this is going to have on infections. There's a very, very good study come out of the States fairly recently, which has started to map the clinical effectiveness of this drug in real world. But the infection impacts haven't yet been reported. And I think that if we could predict what they're going to show, I strongly feel that patients like Angelo, who've already got chronic bacterial infection, will probably rarely actually eradicate that infection. We might find that children are less likely to acquire infections, but I think we've got a long way to go until we know that for certain. And this is why I'm worried. If we look back just over 10 years ago at the age distribution of the UK population with cystic fibrosis, you can see this really big bulk of children in the bottom half of this graph. Some people were living into early adulthood, and then the numbers really disappeared quite fast. Very, very gratifyingly, improvements in healthcare mean that we now have a much, much bigger proportion of adults living with cystic fibrosis. In many countries, this is now more than half of the population. The median age hasn't gone up an awful lot, but you can see this big bulk of patients here in adulthood. And these are people who will only have begun to encounter these modulator drugs once they're already adolescents or adults and they've got fixed lung disease with bacterial infections. And you can see here that the sort of issues that we're concerned about are very prevalent in the CF population. This is just lung infections in 2021 taken from the UK patient registry, and in particular the organisms we're going to talk about today. So chronic or intermittent Pseudomonas aeruginosa accounts for a very, very high burden in this population. Non-tuberculous mycobacteria are less prevalent, but they carry with them enormous disease consequences. And I'll talk a little bit more about why that's so important later on. So there is a lot of recognition that this is a very, very important field, and the CF Foundation, which funds a number of different approaches to treatment, right through from targeting the basic defect with gene therapy or modulators through to infection and inflammation, has got a very good resource that one can look up and see at what stages are these various different approaches that people are trying. And you can see from this that there's a very, very wide range of different antimicrobials that people are trying to develop and that are currently in the clinic. But I think even more encouragingly, there's a very wide range of things being looked at preclinically. And we actually have a CF Trust funded strategic research centre, which is looking particularly at new therapies for cystic fibrosis and mapping onto some of these priorities, for example, donor molecules of NO, repurposing drugs and antimicrobial resistance breakers. So there's a lot of activity in this field, but as you know, many of these products won't actually translate into clinical benefits. We certainly need to enrich this field more. So when we're thinking then about the development of new antimicrobials, as part of the CF AMR Syndicate, we really focused on this unmet need and decided that there was a lack of target product profiles to facilitate a structured approach to develop these new antimicrobials. We wanted to think about building enabling tools and resources for antimicrobial development, and we'll talk about those a little bit in the next few slides. I think many of you on this webinar will be very familiar with what target product profiles are, but they're used as drug development planning tools to provide a regulatory context to drug development, for example, alignment to regulatory submissions documents, and also as a guidance document aligns to unmet medical need and R&D projects. So the process really began with a focus group involving people with cystic fibrosis and their carers, and this enabled the identification of their unmet needs and priorities. These insights were then further reinforced by focused engagement activities with clinical and industry expertise, and together the overall output was a draft of potential TPPs. We wanted really to gain a lot of consensus and feedback on these TPPs. So next came a Delphi survey, and this was conducted internationally and received 85 responses from a range of respondents, including people living with CF and their carers, medical personnel, pharmaceutical industry, microbiologists, etc. And this really demonstrated that there was a very good consensus from the community across the TPPs. Most people agreeing or strongly agreeing to the questions asked. And then the questions, both those which had reached a consensus and those which hadn't, were discussed in further detail in a virtual symposium with leaders in the field interacting with people with CF and other stakeholders. And this is just a comment from Angelo, who we've just heard from, about how being involved in the meeting to identify TPPs was extremely helpful to him as a patient. And Lorna, who works closely in this field and has a child with cystic fibrosis, I think really enunciated the hopes of a lot of us that these TPPs may influence the development of future treatments and make CF easier to manage and more tolerable. So the final outcome following the virtual symposium was the formation of a TPP document, which has three main themes. We focused particularly on the two troublesome organisms, which I highlighted and I'll talk about in the next couple of slides. And I'd like to also, though, highlight the fact that these TPPs will be relevant to other pathogens, and many of them are listed here and are associated with emerging and increasing frequency in cystic fibrosis, but also that cystic fibrosis, I think once again here, is acting as a pathfinder for other chronic suppurative lung diseases. So many patients with bronchiectasis or primary ciliary dyskinesia, they suffer from these sorts of infections as well. So what we're going to actually provide as a resource here may ultimately be very useful for people with broader types of lung diseases. So as I mentioned, where we started really was that the central tenet of the whole process was capturing the needs and priorities of people living with CF for any new antimicrobial, and to develop a very patient-focused TPP. The things of particular concern are the burden of healthcare. Those of you who don't work in cystic fibrosis may be surprised to hear that People like Angelo will spend about 2+ hours every day doing their cystic fibrosis treatments, even when they're well and they look as well as he did on that video. And that sort of burden of treatment will be interspersed by episodes of being less well, admissions to hospital, etc. And all of this impacts on their quality of life. In addition to which, there are adverse effects from the multiple medicines that they need to take, and adverse effects from the stability of their health, because these episodes of exacerbation and hospitalization make it very, very difficult for people to make plans and live their lives as we would all like to. I'd like just to draw attention here that there are specific details on the CF AMR Syndicate website of the published TPPs, so please go and have a look at those. But the key characteristics upon which we focused were the patient population, the treatment duration and route of administration alongside the regime, polypharmacy considerations and particularly drug-drug interactions, how treatment should be monitored, and the risks and potential side effects, because you'll be aware that many antimicrobials carry very substantial risks associated with them. And then also more practical issues such as trial endpoints and storage. So in brief then about these two particular areas of infection focus, the one for Pseudomonas aeruginosa focused really on two main areas. The first is eradication and or suppression of chronic infection, and the second for treatment of acute pulmonary exacerbations in people who are chronically infected with Pseudomonas aeruginosa. And really thinking that the ideal product should have very broad activity against the majority of clinical isolates, including those which are multi-drug resistant, demonstrable ability to break existing intrinsic or acquired resistant mechanisms, because we know Pseudomonas is absolutely very, very well adapted to becoming resistant to any new drugs. No significant pre-existing resistance in clinical isolates, and evidence of synergies with other standards of care, if at all possible, to reduce resistance emergence. And in trying to support these issues, one of the things that the AMR Syndicate has also done, which Paula mentioned, is bring together the community to generate relevant clinical panels of organisms, which people in the community can request to access and can then test their agents on really relevant organisms. Mycobacterium abscessus had three main areas of interest for the TPPs: eradication of infection, which would be a major, major clinical bonus because many people don't ever eradicate this. Maintenance treatment for chronic suppression, which can be very burdensome for people, and again, treatment for pulmonary exacerbations. And the ideal product in this case should really have efficacy against one or more species of Mycobacterium abscessus complex in particular, as this is the one that is most complex in terms of clinical management. Again, ability to break existing resistance mechanisms and no pre-existing resistance profile, and wherever possible, evidence of synergies with other standards of care. Once again, I'd like to draw your attention to the website where you can see specific details of these. So in summary, the TPPs were designed as a co-partner exercise with the community to help guide drug discovery and focus efforts where they're needed the most, based very much on this patient unmet need and the lack of current treatment options. And as such, we think that the TPPs will form a core part of the collaborative discovery funding call alignment to medical need. But thank you for your attention. I'll hand back to Katy.
Great, Jane, that was fantastic setting the scene for us there. And so thanking Jane, I'm now going to move over and ask Neill Gingles and Ed McIver to give us some more details on the Syndicate Collaborative Discovery Programme funding call. Thank you.
Hello everyone, I am Dr. Neill Gingles, member of the CF AMR Syndicate Management Team and a partnership manager at MDC with a background in antimicrobial drug discovery. I am responsible for the Syndicate Collaborative Discovery Programme together with Dr. Ed McIver. Today, I will provide you with an overview of the funding call we are launching, as well as key features of the programme. The Collaborative Discovery Programme, or CDP for short, is a new funding call on our first for the CF AMR Syndicate. Its purpose is to create and advance an exciting and diverse pipeline of preclinical phase antimicrobial projects aimed at treating lung infections in people living with SIDA. The number of projects funded will depend on the quality and stage of the proposal, but the intent is to have selected between 3 to 5 projects to fund and support by the end of 2023, with an overall fund value of £3 million. The main aim of the programme is to provide participants with not only funding, but support to secure delivery of key data aligned with onward drug development and further investment. With this in mind, the portfolio projects selected in the programme will be actively managed. Projects will have clear milestones and respective deliverables, and will receive drug discovery advice and project management input from the syndicate to help successful delivery. What projects are we looking for? Well, our focus of the programme is on unmet needs we defined during our drafting publication of our patient-focused therapeutic target product profiles, or TPPs, for cystic fibrosis-related infections. Just to reiterate from Professor Davies' presentation, we will prioritise projects aligned at treating infections associated with CF, including emerging pathogens, as highlighted in the TPPs. These include proposals which directly act against Pseudomonas aeruginosa, Mycobacterium abscessus. Both these pathogens, if left untreated, often progress to chronic infection associated with worsening lung function and mortality. Other bacterial species, including additional non-tuberculosis mycobacterial NTMs and fungal pathogens, namely Aspergillus and Candida albicans, are also important pathogens, and novel therapeutics related to these infections are in scope for this funding call. What we are looking for, or the funding call scope criteria, is outlined in this slide as an overview. The funding programme is aimed at early stage drug development within HIT validation up to preclinical drug discovery phase, but excluding pre-investigational new drug enabling studies, INDs enabling studies. Within this early phase, projects are at HIT validation stage, HIT to lead optimization, candidate selection, early preclinical studies are all in scope for the CDP, or of note, the equivalent stage in another modality such as biologics. We are looking for novel targets or novel chemical classes, and these can be aligned, including but not limited to the following mechanisms, such as direct acting or non-direct acting antimicrobials, antivirulence approaches, post-immune modulation, potentiators and modulators of microbiota. We are agnostic with therapeutic modality that can enter the programme, which can include, but not necessarily limited to, small molecules, biologics such as antibodies, peptides, phage, and other focused modalities on key prioritised pathogens. We encourage you to talk to us if you think there is a clear rationale for your project to be in scope. Further details of the way to contact us will be covered later in the presentation. I'll run through what's in scope and now what's out of scope for this funding call. We are not looking for projects or supporting proposals aligned with purely target and hit identification, such as in silico or lab-based screening approaches to generate novel targets and hits alone. Any projects in this early phase without clear targets or not within hit validation will not be eligible for this funding programme. At the other end of the spectrum, we are not supporting proposals that are entering investigational new drug or IND enabling studies, such as GLP toxicity and safety studies. Also out of scope are clinical studies, such as first time in human and beyond. Finally, please note we are not supporting projects that are not directly focused on antimicrobial development. These would include proposals related to developing or validating diagnostics and biomarkers or delivery methods alone. As I discussed in the last slide, please reach out to us if you're unsure or think there is a clear rationale for your project to be in scope. Key eligibility for the funding call: we welcome applicants globally, and these can be from academic institutes for academics or researchers from small, medium enterprises or SMEs. The application should come from a single lead applicant. You can have co-applicants, but this is not a requirement with this funding call. The project itself should be planned to last up to a maximum of 2 years in length. It will be required to be milestone driven, with a clear go, no-go progression criteria defined and agreed before the start of the project. The funding in this call contains eligible project costs with the £500,000 in British pounds sterling associated with direct costs, and I will come back to discuss further detail about these eligible costs and a brief summary of the terms and conditions in later slides. As highlighted at the introduction to the funding call at the start of my presentation, our intention is not just to fund, but to add additional collaborative support to enable the best chances of success for projects to attract further investment. The specific requirements will be bespoke dependent on the requirements of the applicant and project, and these will be defined fully during the full application stage of the programme. But the intention really is for the syndicate to support during the project journey, and this may be in the form of drug discovery and disease area insight and advice, support to develop actionable project plans aligned to the TBPs, drug discovery capabilities through the resources of the managing partners, syndicate enabling resources and the wider discovery services network, commercial know-how such as pitch deck preparation, facilitating involvement from people living with CF through the CF Trust, connections across the syndicate CF AMR network, and potential access to downstream funders. You can find more on our website about the type of support we can offer, and we encourage you to discuss your application with us, where we can provide further information about the type of support we can offer and what you may require. So in summary, I've outlined the key facts of the Collaborative Discovery Programme, or CDP. I will end my slide set the summary of the key terms and conditions of the programme. Please visit our website for the full details of T's and C's and see the link below or access this via the QR code. Of note, around intellectual property and the ownership results in revenue share, the funding offer is non-dilutive, and any IP, background IP or otherwise, will remain with the recipient. As the funding will arise from a UK charity, in line with common practice, a revenue share clause will apply upon successful commercialisation, but this will be agreed on a case-by-case basis and will be in line with the Association of Medical Research Charities' current guidelines at the time of consideration. Regards funding, eligible costs up to £500,000 limit will be used to cover direct costs, and typically this could be an appropriate percentage of FTEs of staff plus consumables, and this must be directly related to funded work, access to specialist equipment and reagents, contract research organisation work, use of centralised platforms enabled by the CF AMR Syndicate, and defined lab activities at the Medicines Discovery Catapult or LifeArc. Receiving funding during the project journey will be linked to achieving agreed milestones. But of note, some payments could be made in advance, but will depend on a case-by-case basis. Any questions at this point, please add to the chat and question box, and we will answer in the Q&A session later. I thank you for your attention, and I'll hand over to the next presentation.
Hello, I'm Ed McIver, a member of the CF AMR Syndicate Management Team and a Programme Leader from LifeArc. So today I'm going to outline some further details around the application process and project selection criteria before we move into some Q&As. So from today, the expression of interest is open for applications, and this is done by completing the online form, which I'll talk about in a minute. So applicants will have the option of booking a call with a scientific lead from the syndicate team prior to submission of their form. And it's really important that the deadline for the submission is 23:59 British Summer Time on the 9th of May. So after this, applications will be triaged by an internal panel made up of scientists from across LifeArc, Medicines Discovery Catapult, and the CF Trust. And at this point, we will look to select up to 10 proposals to progress through to the full application process. So in terms of the full application process, the 10 successful proposals, the applicants will be invited to attend an online workshop, which is due to be held in late June. And you will also be assigned a syndicate mentor, which will provide you with help and guidance throughout the application process. And in terms of the deadline for the full application submission, that is the 28th of July. So in terms of the full application review process, this will involve further due diligence by the syndicate teams, and there will be a final review by a panel made up of internal scientists, external key opinion leaders, as well as people living with cystic fibrosis. And we'll be looking to offer funding for between 3 and 5 projects, and we hope to notify successful applicants in early October. And then in the autumn, we will then move through to contract workup, where we'll finalize project plans, budgeting and resource requirements, and all being well, we will be in a position to launch projects in early 2024. I should also say that applicants who are unsuccessful at any stage of the process will be given bespoke feedback with recommendations on how to progress their program forward. So a little bit more about the EOI form then. So it is a relatively short online form requesting non-confidential information only. Now, of course, any information that is provided will of course be treated sensitively. However, it's important that applicants are aware that we will not be under any official CDA at this point. So the online form will provide further guidance around each section, and you'll notice that certain questions are limited to a maximum word count as well. So I'm now going to give a little bit more detail about the types of projects that we are interested in. So since the 1980s, no genuine novel classes of antibiotics have been discovered. And although there have been drugs approved, the vast majority of these target existing mechanisms. And these will face the same issues of emerging resistance. In fact, we were at an AMR conference in Basel recently, and a clinician reported a rather depressing case of an untreatable MDR infection, which was resistant to treatments of last resort, including cefiderocol, which was recently approved. And this really does highlight the urgent need to improve the pipeline of treatments, ideally with completely new acting antibiotics, completely novel. And this is despite considerable efforts in the pharmaceutical industry to identify novel antimicrobial agents. So both GSK and AZ ran HTS campaigns against over 60 essential bacterial genes. And out of these, relatively few leads were identified. However, none of these approaches delivered any new antimicrobial drugs. Now, of course, there are several reasons why these approaches fail, but it is important to learn from from these previous lessons when designing approaches moving forward. So, for example, HTS libraries are generally designed to target orally acting compounds, targeting human genes. And however, we know that the physicochemical properties required for bacterial accumulation are very, very different to typical HTS libraries. It's also important to have an understanding of the level of target engagement required to drive the phenotypic effect in bugs. Do you have to knock it down 25%, or do you have to knock it down sort of greater than 99%? That really will impact the outcome of that particular approach. And in the case of intracellular targets, I think the ability to monitor compound permeability early on in the project and enabling that to be monitored throughout the optimization process should increase the likelihood of a successful outcome. Now, of course, any effective drug will need to reach the target in sufficient concentration and for sufficient duration following the chosen route of administration, whilst avoiding any unwanted side effects. Now, this is hard to achieve for any drug discovery program, but particularly so for antimicrobials due to the additional challenges, including bacterial permeability, the need for significantly higher doses than for many drugs targeting human genes, which of course results in more unwanted side effects. And given that a lot of patients, particularly cystic fibrosis patients, are in rather complex sort of cocktails of medications, it is really important to consider drug-drug interactions, as part of the target product profile. Also, clinical trial design and cost of goods are particularly important considerations, given that for this particular indication, patients might be dosed chronically, and running clinical trials, particularly in respiratory medicine, is very complex. And I think it's important to know that successful applicants of this call must have a credible scientific progression plan, all the way through to final sort of drug development. That said, though, I mean, we are seeing lots of promise, such as novel assay techniques, which enable measurement of compound accumulation. Furthermore, total diversification of approaches well beyond broad spectrum antibiotics. So this would include, for example, host and virulence targets, which could avoid the need for bacterial accumulation altogether. And of course, non-traditional modalities such as phage. And I think the possibility of using AI-based approaches, it could also become a very important part of the design process, because this could help, for example, avoid undesirable toxicity through off-target activity. So I'm now going to talk about some of the key criteria for project selection. Now, as we're inviting applications covering a broad range of modalities, all the way from hit validation through to early preclinical development, we're not expecting every single criteria on this slide to be met. However, I would encourage applicants to consider these very carefully when submitting their EOI to see what aspects of their project does align to these criteria here. So of course a key consideration is how well is the proposal aligned to the CF Syndicate therapeutic TPPs already discussed. Novelty and freedom to operate are also very important. So how does your project differentiate from drugs that are already on the market or in preclinical development? What mechanistic validation is available to build confidence in the approach? What reagents and tools are available, such as proteins, probe compounds, screening assays, or protein crystallography? How likely is the target to be druggable using the proposed modality? And then, of course, there's the collaboration itself. So what capabilities and technologies do you bring to the collaboration? And of course, what additional platform access from the syndicate or otherwise are you looking for? And then finally, there's the progression plan. So what we're looking for is a credible plan which will strongly position the asset to be able to secure follow-on funding and progress towards the clinic. So on that note, here is an overview of the typical data packages that we would hope to achieve at the end of the project, which should make your asset well positioned to secure follow-on funding. Now, this is very much based on criteria required for entry into the Carbex portfolio at various phases. So hopefully we've given you a good overview of this funding call, which we really believe to be an exciting opportunity to collaborate on innovative drug discovery projects targeting AMR, affecting people living with cystic fibrosis. So if you are interested in submitting an application, please visit our website using the QR code on this slide, where you'll find useful resources such as the frequently asked questions, terms and conditions of the call, the therapeutic TPP document, as well as a recording of this webinar. There'll also be the option to book a call with some scientists from the CF AMR Syndicate to discuss your application prior to the EOI submission. And we'll also be attending the ECMID conference in Copenhagen next month. So for those of you who are attending this conference, please come and visit our stand. So finally, the deadline for the EOI submission is 23:59 British Summer Time on the 9th of May. And it really is a key requirement that all applicants who wish to be considered for this call must submit their online EOI using this process. So this concludes the presentation, and I will now hand over to Katy, who will open up the session for Q&As.
Thank you very much, Ed. So yes, we now have about 10 minutes and we can answer some of the Q&As, the questions that have been coming through in the Q&A chat. So please do put your questions in there. If we don't get round to answering them today now, we will be answering them on the website. So don't think that we will ignore those. And I'm going to kick off, with a question to Ed, actually. So the question, Ed, is around the hits. And in this particular case, there's a hit from virtual screen. Would that be considered as part of this funding mechanism?
Okay, so let me answer that. So yeah, I mean, although virtual screening is a perfectly valid hit generation technique, I think for this particular call, we would ideally like to see further validation in appropriate biological assays relevant to CF pathogens to demonstrate that these hits are real and validated.
Great, thanks very much for that. A question, Neill, perhaps you can answer this. How can the CF community engage with this programme?
Yeah, we're keen in terms of the focus on the syndicate is around the unmet needs and the priorities of people with living with CF. And again, in terms of the TPPs that are directed towards that, and again, the funding call is steered towards the TPPs. So we actively encourage the community, and in terms of interaction with people living with CF and the community during the programme, will be an integral part of our programme. And we very much appreciate people looking to get involved in this. Thank you.
Thanks, Neill. So I'm going to pose this question to Ed. A question here that's come in is, if a project developing an NCE that overcomes resistance to a pathogen, but is based on a known mechanism of action and has a known antibiotic class, is the project still eligible to apply, and would you encourage an application?
Yeah, I think that's a very interesting question. And I mean, to be honest, I think what I would say is potentially it would be of interest, but I think you would have to come across in the EOI that how your approach differentiates itself from existing modalities and antibiotics targeting that particular mechanism. If there's a clear sort of advantage, and you can get that across, it's definitely worth at least having an initial meeting with us just to discuss it in a little bit more detail. But I think what I would say is, if we're just looking at sort of a next generation beta-lactam, just with adding a methyl group somewhere, then I think that space is probably pretty crowded, if I'm honest. So that might not be the sort of thing that we're looking for, but I think we'd need to know more to definitely give you an answer on that, to be honest.
Thanks, Ed. That's great. And then Neill, a question for you that's come through on the chat. What are the expected outputs of any project that's selected? Is it completion of discovery, hit to lead, lead optimization, IND enabling?
So again, it would be bespoke in terms of the project and requirements. So depending on where they enter into the project and how they would come out. Again, I would encourage people to come and discuss their projects with us. Again, this will be live on the website. You can actually book a meeting with ourselves to look at how you, at what point would you engage and into the program. So again, it would depend around the project and where it's entering.
Great, thanks, Neill. Then another question around hits or leads, and Ed, I'm going to pose this one to you. So in this particular case, someone has a lead that they've been positioning in a UTI program, and so have been testing it against non-CF pathogens. Would this be in scope? How could we work with this group?
I think obviously, in terms, there's two scenarios. I mean, if there's absolutely no data whatsoever sort of indicating that the new chemical entity may be of have efficacy in CF pathogens, then I think, to be honest, we would want to see some data like that before really progressing. On the other hand, if you did have data showing, let's say, MICs in, for example, Pseudomonas or MTMs, then I think for sure that's something that we'd definitely be interested in. But providing that the package of work that we would be funding is solely geared towards progressing this, the asset in CF patients, so it might be running, for example, some in vivo models, in PKPD models, in sort of CF relevant lung infection models, or sort of extensive microbiology testing in wider CF pathogens. But I think before engaging, we would want to see some sort of evidence, microbiological evidence at least, that that the compounds work in relevant pathogens.
Thanks for that. And now, Neill, perhaps you could answer this question. Where can we find a list of CF relevant pathogens as referred to in the Syndicate biorepository, and how do we go about accessing that?
Yeah, so in terms of the details of the biorepository, if you go onto the website, there's actually a section which will highlight, and there's actually a frequently asked questions section on that. So I'd refer to people to the website on that.
Great, thank you. So the practical question here, Ed, maybe you can have this one. Can an investigator or an applicant be on multiple applications?
I'd need sort of more clarification on what you mean by multiple applications. I'm assuming it would be multiple applications with with the syndicate member, if that's how if I've understood the question correctly. I mean, there's nothing ruling that out, but clearly, so long as there's no sort of duplication of funding from this call into work packages associated with other applications. But I think that, and again, is a case-by-case scenario where we would need to, I'd strongly encourage you to book a call with us and, and discuss that in more detail.
Thanks. Neill, a question here on IP. Who will own the IP generated in a funded project, and do they need to have patents in place before applying?
Thanks, Katy. No, if there is background IP coming into the project, that would be with the recipient. Again, if there is IP generated during the project, that would remain with the recipient or the applicant. Again, if they don't have any IP, it would be looking at, in terms of the application, that there's an actually exploitation route and there's actual freedom to operate. So really looking at whether they can actually generate that. That answers the question.
Yeah, that's great. Thank you very much. Another question here, Ed. Is there a requirement for novel mechanism of action? Does the target have to be completely new?
I don't think it has to be completely new. I mean, what I would say is, though, if it was, say, a novel modality or novel approach to an existing mechanism, which could potentially overcome, for example, established resistance mechanisms, then absolutely, that's the sort of project that would be very much interested in, because there'd be an inherent degree of validation and confidence, you know, with that, you know, particularly if it's a target that's already got, you know, drugs on the market for. So no, it's absolutely not a requirement, but obviously Me Too's, you know, fast followers, you know, same approaches targeting, you know, existing, you know, mechanisms, you know, that's probably going to be of less interest to us. But again, come and talk to us and we'll discuss it in more detail.
Great, thanks for that. And then Neill, another question on who can apply. And the question here is, can a consortium apply?
Yes, in terms there can be a single lead applicant or co-applicants. Again, this can be international. It would obviously depend, but what it would be the lead applicant that have to actually submit it. But it's not a requirement to have a consortium or multiple partners. Okay,
thanks. Ed, are non-traditional approaches in scope including host targeting and antivirulence?
Yeah, both of those very much are in scope, and you know, as was indicated in the slides, and you know, again, yes, we're very interested in hearing from applicants with those approaches.
Great, that's really good. I think we've probably got time for one more question, and I think that question is going to be around. Oh gosh, what have we got? Oh yes, for the progression plan, how downstream or detailed does it need to be at this stage? And perhaps Neill, you can pick that one up. So how much detail do we need in the progression plan?
Yeah, again, it'd be bespoke for the project. So again, it would get the person to reach out to us, again, book a meeting. In terms of the EOI form, when you see it on the submission, it's a small form, so there will be opportunities to further work on the full proposal. But again, I would probably emphasize just reach out and actually we can talk through that project plan.
Yeah, that's great. Thank you. So I think with that, and I appreciate there are a few questions still in the chat. We will get back to you on those. I'm going to close out this session, but thank you very much for joining us. I hope you found it informative. I would encourage you, please, to look at the website for more information and the updated frequently asked questions. And what I can say is that the call for expression of interest is now live and it is open. The website has been launched. We will make available a recording of this webinar and of the Q&A session, so you will be able to access those and you will be able to start to book a phone call and a meeting with us. So thank you very much for joining us once again. Thank you to Jane, to Angelo, Paula, Ed and Neill for your participation too.